Hello? Can you hear me well? Yeah. Okay, good afternoon, everybody. So the sugar level is up again. So today I will be talking about epigenetic reprogramming of aging, or ERA, also known as transient reprogramming, but I prefer to call it ERA for reasons that will be clear in a minute. So ERA was developed and patented in my laboratory at Stanford, where I'm an assistant professor, and then was licensed exclusively to turn.bio, of which I'm a co-founder shareholder, a company whose mission is really to translate this technology into people. So the nuts and bolts, the details of the science behind this are publicly available. So we just posted a few days ago the paper online on bioRxiv. So all the details are there, and today I will just quickly go through them. So I want to start actually from one of the questions that Mike West had yesterday. So what is the unifying theme of aging? Well, my vision and my opinion is that the unifying theme of aging is epigenetics. As when we are young, the epigenetic, which is the program by which cells know what to express and what to not express, so what defines the cell identity in a way, is very tightly controlled at all levels, okay? As we aged, this becomes dysfunctional at all levels. So the question is, this basically results in two different outcomes. So the first one, when epigenetic damage is also accompanied by genetic damage, for example, telomere attrition, you have a cell that becomes senescent. So it enters replicative senescence, and it stops dividing, and it starts spitting out bad stuff, the SASP. Now, with senolytics, we can go in, kill the cell, and release and reduce the SASP. But the problem is that the senescent cells in the body, in the aged body, is only 1% of the cells, okay? So the rest of the cells do not experience senescence, but they still age because they go through epigenetic damage. So the question is, can we reset this epigenetic landscape to a more youthful state? Can we put it back where it belonged to before? And the answer is yes. Nature does this, since billions of years, okay? So through reproduction, always resets the age back to zero. And the same process can actually be feasible in vitro through the process of IPS reprogramming. But Houston, we have a problem in the sense that when we do this resetting of age through IPS, we also change cell identity, and that's something we don't want because it's potentially tumorigenic. So 5 years ago, Jay, you probably remember him from last year, very talented, a brilliant mind, came to me and he said, look, Vittorio, I want to study aging because I'm profoundly interested in aging, and I have a question, I have an hypothesis. What if by transiently expressing the reprogramming factors, what if we can decouple the two processes and achieve cellular rejuvenation without affecting at all cellular identity? And I remember my reaction to that, which was pretty much like this, was like, as visionary and crazy as it sounds, it could work. That's exactly my expression back then. Now, 5 years later, I'm here to tell you that it not only could work, it does work, okay? And we have shown this. It's a little, what's going on here? Okay, it does work. And so our work shows that we can actually indeed rejuvenate the cells, improve their cellular function, but retain their cellular identity, okay? This is the key concept here. And now we have done it across 7 different cell types, human from elderly people, naturally aged people, okay? We have used a clinically translatable approach based on mRNAs, which will, quickly move us to the clinic. And we have done this way and beyond the hallmarks of aging, okay? We have shown this at the transcriptomic level. So as the cells age, they start expressing different blocks of genes in very apparent ways. With our treatment, we see that actually those genes that change with age are actually reset in a more youthful fashion. We have proven this at the methylation clock level. So we looked at the Horvath clock, and we see that we can get rejuvenation all the way up to 5 to 10 years in terms of quote-unquote methylation age. We have shown this at all the physiological markers of aging. And we have seen that for those markers that go down with aging, they actually increase with our treatment. The ones that go up with aging, they decrease with our treatment. The only thing that doesn't change is telomere length. Now, many people look at this as a very bad result, and actually I'm very happy about this result. Because remember, the claim is that we're not changing cell identity here, okay? If we were to see telomere elongation, that would speak for TERT activation, which means change in cell identity and potential tumorigenesis, okay? So I'm very happy to see this. But then, of course, one marker that we could not, one hallmark that we could not test in this context, because these were fully differentiated cells, was stem cell exhaustion. So now imagine just for a second that we can rejuvenate stem cells. Now this has dramatic implications, okay, for regenerative medicine, because stem cells are naturally endowed with this ability to self-renew and to regenerate an entire tissue. And so I reached out to Marco, who was back then a postdoc in Tom Rando's lab. Now he is an independent investigator and the director of the Rejuvenative Center. And I knew he was the man because a few years back, he actually developed a technology to mimic in vitro the micro niche that the stem cells, the muscle stem cells, have in vivo. So he was able actually to keep the stem cells quiescent in vitro, which is fundamental here. And I said, Marco, can we actually do the treatment on your cells, not your cells, but on the muscle cells, and can we see if there is any rejuvenative effect? And that's what we did. So here you see the untreated ones, the treated ones. They were transplanted into a young host immunocompromised mouse, but before then they were tagged with luciferase and GFP to longitudinally follow them. And then they were transplanted into the tibialis anterior, so a muscle. And at the time of transplantation, we also injured the mouse, the muscle, to kind of challenge the cells to regenerate the muscle. And then we followed them for 11 days, and a second cohort was actually injured a second time 60 days after. And here's what we got. So we transplanted the untreated cells here and the treated ones in the same mouse contralaterally to have the same environment. And first of all, we saw that the regenerative capacity of the rejuvenated ones, which are the ones that you see here in cyan, were comparable to the regenerative capacity of young ones and much superior to the old ones, untreated, that you see down here in red. We did morphometric analysis, and we could see that actually we could see more fibers and thicker fibers, and this was quantified here, to levels comparable or even superior to young, untreated muscle stem cells. And then, very surprisingly, we saw that just to test if there was homeostasis and memory of this process, 60 days after, so 60 days after, 2 months, in the lifespan of the mouse, this equals to about 6 to 10 years in the lifespan of a human being, okay? So it's a long time. We saw that the rejuvenated cells were absolutely undistinguishable in their regenerative capacity compared to young ones. Then we said, okay, that's not enough. Let's move to the next level. So let's transplant now the stem cells into an aged environment to mimic like the nasty, bad environment of an aged muscle, right? We follow same experiments. We follow them for 30 days. And this time we measured tetanic force. So it's an in vitro assay that measures the contractility and the force in vitro. So follow me for a second. So this is the force that you find in a young, untreated mouse. This is the force, the line, the red line, that you find in an aged, untreated mouse. Now, when you transplant an aged mouse with aged cells that have not been treated with our technology, you restore the physiological force of that environment, of that muscle. If you transplant it now with cells that were aged but rejuvenated, you now restore the force of a young mouse. We're talking about 30% force induction, which is the most significant force induction that has never been reported in the literature. And you can see that this is true across all the single data points. But so this these data for the mice in the audience are very promising and very encouraging, but for the human in the audience, I also have got good news. We actually saw the exact same thing. So we isolated muscle stem cells from patients all the way from 10 to 80, 90 years old, and we see the same effect. What's really striking is that we always see a benefit. The problem, the fact that, but this benefit is very minor when we go all the way up to 55 years of age, but it becomes really dramatic when you start dealing with cells that comes from elderly patients, suggesting that there is probably really an epigenetic crash and that the reprogramming is taking care of that. So in summary, ERA promotes cellular rejuvenation. It enhances the regenerative potential of stem cells to youthful levels. It appears to be safe. Our data suggests that there is no change in cell identity and there is no tumorigenesis induced by this approach. The platform is clinically translatable, and this can be broadly applied to a variety of different cells and tissues. So, of course, you have a number of indications already in mind, but, and I want just to conclude by saying that as we age, the cells become epigenetically aged. Some of them are senescent, and we can actually, with senolytics, come in, kill them out, and reduce the SASP. But there is a ton of cells left behind that are still aged. So what do we do? Here's where epigenetic reprogramming of aging comes in place. So our aspirational goal, of course, is to do this in vivo, and we have already proof of principle that we can deliver mRNAs in the eye, in the hypothalamus, and in the muscle. And with that, I want to thank, of course, the academic side of this project. I want to really thank the M Fund and the Methuselah Foundation for the initial funding in the company, Gary Hudson, our CEO, Sergio, our COO. And while I take your questions, I want just to leave you with a movie that really nicely recapitulates what I have been talking about for the past 10 minutes in a much, much nicer way. And thank you for your attention.
Thank you, Vittorio. That was really good. And I'm sure there'll be plenty of questions. So start over here.
Oh, thank you, Vittorio. Thank you very much for a great presentation. And it really warms my heart to see epigenetic rejuvenation approach finally get recognized for the potential it has. One question, what do you see a potential therapy for humans? How do you see it with mRNAs? And if you see any other type of gene delivery for the factors, maybe for systemic rejuvenation and not just ex vivo?
Yeah. So by nature, I'm the half-full cup guy, so I actually see a lot of potential here, and I think that the potential is unlimited, quite honestly. But I don't want to overestimate it, okay? Well, our data suggests strongly that, in the short term, we could potentially apply this to osteoarthritis, sarcopenia, and skin aging. And then there is a number of other data actually that are slowly cooking that actually are opening up to other applications down the line. Regarding the in vivo delivery, so we are partnering up with Gary and his company that has developed a cutting-edge development in vivo delivery method. So we are actually kind of joining forces, and we think that this could be absolutely a winning strategy. And we're thinking about, of course, in tissue-specific delivery, and one day even systemic delivery.
Great, and we've got a question just here.
Yeah.
Thank you very much for the talk. So we heard yesterday about retroviruses and activation with aging of these retroviruses. So did you study how your reprogramming affect activity of these endogenous viruses?
Yeah, we have not looked at that yet again, but another side of the project in my lab actually has been looking at the reactivation of retroviral elements. And we know that actually they come up and become deregulated with aging, but the factors that we're dealing with, which are, of course, embryonic factors, they actually have binding sites for these elements. And my feeling, and I have some data in support of that, is that they actually can go there and also silence them properly.
Have you got any more questions?
Yeah, so we already talked to each other yesterday a little bit about this, but I was just wondering if you'd share with the rest of us what your timeline is for the application of this technique, right?
Yeah, So we strongly believe, and we're absolutely optimistic about that we should be able to actually get to the IND and to early clinical phase within 2 to 3 years.
Is anyone just down here?
You said that you didn't observe any tumorigenesis or a loss of cell identity. However, I guess the mice have not died yet, so can you actually conclude they won't get cancer a little bit later?
So that's a great question. So, well, the mice were already 20 to 24 months old, so they died, but at some point, but just because they were too aged. But we actually followed them up for 6 months, and in 6 months we did not see any sign of tumorigenesis, teratomas, cysts, dysplasia, nothing, just fibers. So so for the mice we've used 20 to 24 months old donors. For the humans we went all the way from 10 to 80, 85 years old.
Great. Okay. Have we got any more?
I don't know.
What do you take as a readout to assess for how long you can treat your stem cells before they're in the state that you can give them?
Yeah, that's a tough one. I mean, it really depends on the stem cell type you're dealing with. Now, if as we did, you can really, tackle or you can really deliver this to stem cells. Stem cells have a lifelong, regenerative capacity. And our data again suggests that, they retain that memory. So my hunch is actually that this can be really, really extended in time. But of course, it depends on the tissue. It depends on the micro-niche.
Excuse me. No, I meant under the treatment.
Yeah.
In order to reprogram them.
Yeah.
How do you decide for how long you do that? What is the?
Sorry, I didn't get that. Yeah, it's actually that is actually part of the really the hard part of it, in the sense that we optimize the treatments in a cell-specific manner. And so, for example, for the stem cells, it was only 2 days of treatment. So this is very rapid. Okay, it's pretty dramatic. It was just 2 days.
Great. Thanks, Vittorio.
Thank you.
Thank you. And